Griffith proposed the idea of DNA could be the genetic material. Still, there was a confusion about whether it is DNA or protein or something else. Later, Hershey and Chase confirmed this hypothesis using a virus. As I told, these viruses require a living host. It could be humans or animals or even a microorganism such as bacteria. Viruses infecting bacteria are called bacteriophages. The bacteriophages used by Hershey and Chase were simple particles composed of only protein and DNA. The outer structure of these phages was made up of a protein coat and the inner structure contained a DNA core. In this experiment, they used E. coli as a host. Once the virus attaches to the host cell, only its genetic material is injected into the host and the viral coat remains outside. The infected bacteria are manipulated by the bacteriophages such that the bacterial cells start to replicate the viral genetic material. Finally, many new virus particles will be produced inside the bacteria, which come out by lysing the host cell. Hershey and Chase wanted to check whether the injected genetic material is protein or a DNA. The experiment was conducted in two separate batches. In the first batch, the viruses were cultured in a medium containing a radioactive isotope of sulfur, that is S35. A radioactive isotope is an unstable form of a chemical element that releases radiation in the form of alpha, beta, and gamma rays. In the experiments, the natural elements are replaced by their isotopes, and the radioactivity produced by these isotopes is measured to prove some hypothesis. So, in this experiment, they have used S35. Sulfur is found in many proteins and is absent in DNA. So, the phages labeled by this method will have radio-labeled proteins. In the second batch, the viruses were cultured in a medium containing a radioactive isotope of phosphorus, that is phosphorus 32. Phosphorus is found in DNA and not in proteins. So, the phages labeled by this method will have radio-labeled DNA. In the next stage, Hershey and Chase allowed these labeled phages to infect bacteria. After infection, the culture was agitated to separate unattached viral coats from the host. Later, the culture was subjected to centrifugation. Centrifugation is a technique that separates the particles from a solution based on the density. Since bacteria are heavier than viruses, they move to the bottom of the tube and form a lump called pellet. Lighter material such as empty phage particles remains near the top of the tube and forms a liquid layer called supernatant. When Hershey and Chase measured the radioactivity in the pellet and supernatant from both the experiments, the radioactivity was observed in the supernatant of the culture infected by S35 virus, whereas the radioactivity was observed in the pellet of the culture infected with P32 virus. This clearly concluded that the protein part stays outside the bacteria and therefore is not the genetic material. In contrast, the DNA has entered the bacteria and instructed the host to produce new virus particles. Therefore, this experiment clearly concluded that DNA is the genetic material and put a full stop to all the confusion related to DNA.